There we go. So thank you very much for joining us today. We're going to cover in the AREC seminar series, which happens on the last Friday of every month. This month, we are going to be talking about Eastern Shore AREC. Thanks to Dr. Mark Ryder, Director of Eastern Shore AREC, for joining us today. It's my understanding we're going to have presentations from a couple other faculty members as well. I'm going to go ahead and turn it over to Mark. At the end of the presentation, we'll have some announcements on events coming up, so stay tuned for that as well. Take it away, Mark.
All right, thank you, Matthew. It's really nice for us to showcase a little bit about the Eastern Shore AREC today. I'm Mark Ryder, and I'm a Soils and Nutrient Management Extension Specialist. I'm located here at the Eastern Shore AREC, and I'm also serving as Director for the Eastern Shore AREC as well. So what the plans are for today is, first I'll give a general overview of the Eastern Shore and all of the research programs we have ongoing here. And then we'll delve a little deeper into the plant pathology and weed science programs. And BJ Singh and Doug Higgins will speak more about those specifically. So with that being said, thank you all for joining us. It's always a good time to talk about things we do and as far as research and extension. Uh-oh, my slides aren't advancing. There we go. So a lot of times, hopefully you all know, you're from Virginia, but when we speak about the Eastern Shore, we always want to make sure people understand where we are specifically, as it's a pretty important agronomic region here in Virginia. And as you know, Virginia is a very diverse state. So the Eastern Shore, you know, Blacksburg's, many of you are joining us from there, out in Southwest Virginia. We're about a 6-hour drive east, located on the lower Delmarva Peninsula, where Virginia has a very diverse agricultural system, but we also have a pretty diverse system here locally on the Eastern Shore as well, where we raise anything from fresh market tomatoes, potatoes, snap beans, to things such as poultry. We're the one of the top broiler counties in Virginia here in Accomack County. So we have the poultry litter associated with that to utilize as well, but also traditional grain crops such as corn, soybeans, and wheat. So due to this diverse system, hopefully you've seen this map before and are well accustomed to seeing this map, as we have many different ARECs around the Commonwealth that focus on different things that are important to that area. So here we are on the Eastern Shore that focus on things specifically important to the Eastern Shore, but also to Eastern Virginia. The entire Eastern coastal plain has very similar agronomic systems. And so we can easily translate our research to other parts of the Commonwealth as well. Just zoning in a little to show you our specific region. One of the things that's very important, near and dear to us, is our location as it's related to the Chesapeake Bay. So this is the Chesapeake Bay, which is one of the largest estuaries in the United States. And it's very important both environmentally, but also culturally, as far as historic references, but also for production, as aquaculture is a very important industry in the Commonwealth as well. Zooming in, just via some of aerial satellite imagery, you can see there's fields everywhere. This is a very agriculturally important area, and regardless of where you go here on the Eastern Shore, they're connected to agriculture in some fashion. So certainly agronomics, agriculture, forestry are number one. And then zooming in a little more, this is our field layout of the Eastern Shore AREC. Overall, we have about 220 acres within our AREC direction here. Now, that's one of the smaller ARECs if you talk to some of the other directors around the state. And about 110 of that is farm. So this block is 110 acres that we produce vegetables and grains on. And we're laid out with a central road, and then every 220 feet, we have a side road to establish our plot area. We have underground irrigation on the entire farm, which supplies overhead irrigation. But we also have these first 4 blocks can be irrigated with drip irrigation for high-value specialty crops. We have a pretty unique history, which kind of leads into the next few slides to show, why we are where we are and do what we do. So spanning back to 1897, growers in Southeast Virginia decided that they wanted to form basically a union to do research on crops important to them. So this vegetable growers joined a association called the Southern Produce Company in 1897. And by 1907, they had pretty much a research station established in Hampton Roads that was under the USDA umbrella to do vegetable research. In 1920, they became part of the Virginia Department of Education, who assumed operations, where we continued for many years. In 1912, they decided, hey, there's a lot of vegetable and grain crops being produced on the Eastern Shore, and we're so different from Hampton Roads, they said we really need to establish a research station over there. So by 1913, they leased some land, and we're actually taking a ferry boat across the Chesapeake Bay, which I'm not sure, somebody a little more experienced than me can say how long that took. I don't remember. I wasn't here then. But it takes several hours to commute to the Eastern Shore to conduct research on things important to the Eastern Shore. By 1956, we moved to our current location where the current building was built, which is this main brick building, as well as this outbuilding of the shop and the head house. And in 1985, we actually merged with Virginia Tech. So we were part of the Virginia Department of Education until 1985, when Virginia Tech and this Southern Produce Association signed a memorandum of understanding to join the university. So of course, since 1985, we've been part of Virginia Tech and focusing on research, teaching, and extension. Now, where this timeline continues, we're excited to say is in 1956, the main building was built with our outbuildings. In the mid-70s, we had an addition added on with a classroom, and it's currently where our plant pathology, soils, and entomology labs are located. However, we're going to continue this upgrade and expansion over the next few years, as we have been selected and funded currently for a study to take a look at what needs we have here on the Eastern Shore and at the Eastern Shore AREC to continue our applied research and extension programs. So as you'll see through the remainder of the presentation, we have a large faculty, staff, and graduate student contingency that is really aiming to continue this research. And one of the key parts of that is student housing. So this is one of the main things we hope to improve over this capital improvement plan, along with an AREC support complex, which will aid in our spraying and pesticide storage, a renovation of the main building here of labs and office space, a new education facility where we can have meetings such as Oyster Rose, ag conferences, extension meetings, and also a new smart greenhouse. So in total, we hope to have around 25,000 square feet of new construction and 14,000 square feet of renovation. So this is where we continue to grow over the next 3 years. We're currently searching for an architectural firm and are in negotiations with our working drawings to be complete by December of 2025 and substantial completion and move-in by January of 2028. So that's the goal, where we hope to see the Eastern Shore here in the next few years. Now, with that being said, I know some of our Eastern Shore counterparts are listening. We're already about 2 months behind schedule, so it'll probably be sometime in later 2028. But nonetheless, we are hopefully going to see these improvements in the near future. With all these needs over the last 100-something years and current needs changing, our vision and mission really have not changed. Our overall vision is to ensure an economic and environmental sustainability by building on the land grant mission to serve the Commonwealth and global communities. And we specifically do that related to vegetable, grain, oilseed, and fiber crop production systems. So every time you're in a crop production system, especially here in Eastern Virginia, we really need to pay attention to our land, air, and water resources, since we are in so close proximity to national treasures like the Chesapeake Bay. And of course, through all these activities, we expand our applied research and teaching as it relates to farmers and the industry here in Virginia. More specifically to our local needs, to give you an idea of what's important here on the Eastern Shore, Accomack County currently rates third in farm gate value out of all the counties here in Virginia. And to give you a scale on size, the average farm in Virginia has $140,000 farm gate value. Here in Accomack County, each farm has about $1.3 million in farm gate value. As it relates to vegetables, where we are third in Virginia and 289th in the USA as far as counties. So we're pretty highly ranked within the whole country as far as vegetable production. Third for grains and oilseed, and 10th in cotton. And within Virginia, we are third for livestock, which is pretty much poultry, broilers, and 69th in the USA. As far as livestock, you don't think of livestock being in the water, but we're fourth for aquaculture and 62nd in the USA. So the southern county here on the Eastern Shore is Northampton County, who is 8th in Virginia for cash receipts for farm gate value. The average farm size there is around $724,000 in value. And they rank first in vegetables, notably green beans or snap beans and potatoes. Third in nursery stock. We have several large nurseries in the county. 8th in cotton, and first in Virginia for aquaculture, mainly hard clams and oysters. As far as research specific here to the Eastern Shore AREC, we have a pretty diverse operation due to the diversity of Eastern Virginia agriculture and also here of the Eastern Shore. So every year we have 34 plus crops grown on the station, ranging from high value specialty crops like tomatoes, snap bean, watermelon, sweet corn, to other things such as basil, onions we've had, soybeans, corn, wheat, typical agronomic crops, but also cover crops, which is a pretty large component of many of our research programs right now to see what grows best here in Eastern Virginia in our climatic zone. Regardless of what we grow, what we do, we have this integrated program that we focus on, which is pretty typical of the AREC system here in Virginia, where we take an applied research need, such as cover crops. We figure out the best way to grow these in Virginia agronomically, as well as economically and environmentally. We include the field component, in lab diagnostics or analysis for those more scientific needs, but also integrate smart agriculture into the system as our farmers and industry is moving towards more refined approaches to really enhance agriculture for economical and environmental reasons. And of course, through all of these activities, applied research, whether it's in field, in the lab, we really focus on getting this information out to stakeholders, which we did. This is a fresh slide we just made because we're high off the heels of our Eastern Shore Ag Conference and Trade Show, which happened this week, this Wednesday and Thursday, which is our premier event for extension outreach activities. This is just a chance for 40-something ag industry businesses to speak with our 200 plus producers, as well as our Virginia Tech and Virginia Cooperative Extension faculty, staff, and students to really focus on what we've learned and how to improve agriculture here in Eastern Virginia. So just a few spotlights. Typical speakers at this event are some of our partner institutions, such as the Virginia Department of Agriculture and Consumer Services. Commissioner Joe Guthrie was here speaking. We had some colleagues from Virginia Tech at administration, such as Amy Sebring, who's our chief operating officer, as well as others in leadership levels at the university. And in the past, we've had Provost Clark attend as well, as far as Dean Ferruzzi and Dr. Burrows with Virginia Ag Experiment Station. So it's always a great experience and time for Virginia Tech colleagues to really see what we do on the ground to better explain why Ag Experiment Station is such an important part of Virginia Tech. And of course, we have an oyster roast every year. We have education sessions at the Moose Lodge here in Belhaven. But also education sessions continue over oysters and clams and barbecue in our new shop, well, say new, 5 years ago or 6 years ago or so, shop facility where we can just mingle and have conversations in a more casual setting as well. We also take our extension and research internationally and host events here in Virginia. This past September, the Eastern Shore AREC, in collaboration with Virginia Cooperative Extension, especially Northampton and Accomack counties here on the shore, and Virginia Beach across the bay, we hosted the International Soil Tillage Research Organization meeting, where we showcased Eastern Virginia, Eastern Shore, and pretty much USA agriculture, as we had a soils and tillage and nutrient management conference on the Virginia Beach oceanfront. So overall, we had 211 registered attendees, and we had representatives from 35 registered countries, but really attendees were present from 46 different countries around the world. So this was a great experience to take extension basically on the road here in our back door, so we could showcase what we do in Eastern Virginia to many different cultures and people with many different production system expertise. So more about our research programs here at the Eastern Shore AREC. We currently have 4 tenure-track faculty, which they'll have the opportunity to speak a little more on their programs later. 1 postdoc associate will hopefully be hired here in the next month or so. 7.5 staff that are permanent that work within our AREC. 8 graduate students with, we're recruiting several more right now, so that number should be about 12 here in the near future. And somewhere between 3 and 20 seasonal employees, depending on the time of year, to really get the work done here in our research plots. As far as programs themselves, we currently are searching for a horticultural sciences position. Emmanuel Torres was with us, and he left us in February. But this position is approved, and we're currently working through the hiring process to have someone really focused on the overall efficiency, resiliency, and sustainability of food systems related to commercial vegetable crops. And notably, currently, it'll be plants such as tomatoes, watermelon, broccoli, lettuce. And this person will really focus on not only varieties, but also other agronomic needs, such as how do you grow them as far as irrigation, tillage, insecticides, fungicides, really to bring all of us together to make sure we can grow crops economically and also have high value, high quality crops as well. Dr. Vijay Singh is our weed scientist, along with his graduate students and research specialist, R.C. Cooley. So Vijay is going to talk a little bit later, so I'll let him explain his program. We have a new hire that started on December 25th of 2024, Dr. Kemper Sutton. Some of you may recognize him as his PhD was from Virginia Tech in Dr. Kuhar's lab. And Dr. Sutton will have a field and laboratory component really focus on applied entomology related to high-value specialty crops here on the Eastern Shore, but also the agronomic crops important to our area. Dr. Doug Higgins and Kaylin Adams run our plant pathology lab here on the Eastern Shore, and Doug's going to speak to you, so I'll let him talk more about that as well. And then we have the program focused on soils and nutrient management, which is myself, and also John Mason, who's our research specialist. And we really focus on fertilizer management, cover crops, specifically nutrients that are important environmentally and agronomically, such as phosphorus and nitrogen and potassium, and also developing new recommendations for our growers, such as sulfur. So with that, thank you for your time. Come join us here on the Eastern Shore whenever you are passing through the area or at events such as this photos from the Virginia Ag Expo last year. We always like to meet and speak with those of you from campus and other ARECs as well. So with that, I'm finished. Douglas, if you want to take over the screen and talk a little bit more about plant pathology.
Great, will do. All right. So my name is Doug Higgins, and I'm an assistant professor and extension specialist here at the Eastern Shore AREC. I have a 50% research and 50% extension appointment. And so my lab mainly concentrates on applied research problems in vegetables and small grains. We always hopefully what I hope that you get out of this presentation is how we like to start with applied questions and how those can start to lead us towards more fundamental questions that we might explore. So I'm going to cover today
Hey,
just an
Doug.
overview.
Hey,
Yep.
Doug, sorry, I think we're not seeing your slides.
Oh, you're not?
Yeah.
You just
Shoot.
got to go full screen.
I thought I was full screen. Yep. Let's see.
Will we see your next slide?
So
There you go. You got it, man.
that's it. That's good.
You're good.
Okay, cool. What I'd like to cover today is really just kind of give an overview of the projects that are currently up and going in my lab, and then some of the ones that we got that have got funding for that are just about to get started and rolling, and kind of just give you a taste for what we do here. So let's see. So the first project I like to talk about is a project that I've been working in collaboration with Dr. David Langston over there at TAREC. And we've got a project looking at developing and improving some management strategies for a fungal disease of watermelon called anthracnose. This disease, if you were to see it in the field, it would start as these kind of, spots in the field that has a very patchy distribution at first, and that photo on the left there. And our watermelon growers have really been kind of struggling to get a hold of this one. Not only does it take out, leaf tissue and outright kill the plant, it can get onto the watermelons themselves. This is a picture that we found post-harvest. These watermelons were picked clean in the field and then put into cold storage, and this disease popped up as soon as they went into the cold storage. So it also is causing post-harvest losses. David and I co-advise a master's student on this project, on a portion of this project, Emma, who's pictured up here on the right. And what Emma and I set out and did this season is we ran some basic fungicide efficacy trials because some of our grower group were complaining that some fungicides weren't working very well when they had in the past. And we actually found there was a couple other interesting group of fungicides that appeared to be stopped working. So these are just some pictures I have of up here of kind of what we saw in our first field season. On the left there, we had our non-treated plot, so you can kind of get a sense of how aggressive this disease was. We introduced the pathogen into this row of watermelons just on a single plant in the middle, and it just took off and ran and pretty much clobbered the plot. But we did see that there were some good efficacy with some prothioconazole, and compared to other DMIs on the far right there, which is that methyltrifluconazole. So there were some interesting questions on why some of the DMIs aren't working and others are. And the thiophanate methyl in the middle was of interest too, because traditionally that had been working for growers as well. We actually saw the strobilurin's to work quite well. So this is just kind of an example where we started with a basic field trial looking at product efficacy, and that's kind of launched and helped narrow down the questions that Emma is going to answer. So Emma went around here on the Eastern Shore, up in the Northern Neck, and in Capron area, and surveyed watermelon growers and made a big collection of Colletotrichum obliculare isolates, which is the fungal pathogen that causes the anthracnose disease. So she got those all collected. She got them back into the lab. She has to do some single sporing of them to make them individuals and then put them into long-term storage. And that pretty much took her through last season. So she's starting off right now, and she's going to take a look at a couple based off our field trial. She's going to take a look. One hypothesis we have is that there might be some fungicide resistance. So she's going to screen for 3 different fungicide groups. So she'll phenotype them with the. Oh, go ahead. Yep. Does someone have a question? Yep. Well, if you do have a question, just feel free to jump in there. I'm happy to take them anytime. But Emma is going to phenotype for some fungicide sensitivity to 3 different groups of fungicides. And then she's interested in pairing that with some genotyping to look for these point mutations that are known to be linked to fungicide resistance. And so this will really be the first time that we're taking a look at Colletotricum bulgari in Virginia and really kind of in the Southeast. We're part of a larger group working on this. With Emma, we also structured some of her sampling to answer some questions about temporal and spatial changes in the population structure. So she has some interest in using some of our population genetic tools to answer those questions as well. And that's what she's developing as part of her master's project. The other part of this project that I'm working on, we're currently in recruiting and have been interviewing postdocs positions to come in and really take a look at a little deeper at this pathogen population. One of the feedbacks that we get from our watermelon growers is that they go to buy a variety of watermelon from their seed catalog, and they see it advertised as resistant to anthracnose in the catalog, and then they go put out in the field and man, it just gets hammered. And we saw that in our field plots when we put out a cultivar trial. And so what is happening really is most of the commercially available resistance out there is available to race 1. So the fungal pathogen in this case has kind of 3 different races or forms that it takes. It can, there's 3 different races. So race 1, 2, and 2B. And so most of the cultivar resistance that are commercially available is to race 1. And our hypothesis is that most of the isolates out there aren't race 1, they're race 2. So this postdoc will come in and do some what we call race typing, which are going to utilize greenhouse and growth chambers under controlled conditions and a panel of cucumbers and watermelons to determine what race there is that we have. And we'll be combining our collection and doing the race typing for the Northeast cooperators or kind of mid-Atlantic. So this project will involve, also screening and race typing for Colletotricum isolates collected in Delaware and New York, in addition to Virginia. So the next project I wanted to highlight here is a project by Pooja Panwar, who's a master's student working in plant pathology. She's pictured up there on the right. In Pooja's research, she's interested in improving stands in early planted sweet corn. One of the things that fresh market sweet corn growers love to do is to try to have the first fresh sweet corn at the stand. And to do that, they keep pushing the envelope on wanting to plant early and earlier. The other background piece of this story is that the most popular type of sweet corn is called a super sweet corn. And it's popular with the growers because it maintains that sugar for a little while after you pick it. So it doesn't have, you don't have to eat it the day you pick it. The downside of super sweet corn is that it has a very thin seed coat and it's brittle. And when you put that seed into cold, moist soils early in the springs, it cracks open and it attracts pathogens that kind of come and feed on the metabolite. So what you can do is add a fungicide containing seed treatment to help improve your seed stands. And so what we've been running here is some trials for the International Sweet Corn Development Association, where we trial some commercial standard sweet treatments. And I just put some pictures up here. We've run 2 years of these trials now, and you can see that there's quite a difference when we have no fungicide in our seed treatment. We can see, very poor stands around 30 to 35%, and we can double our stands in those very harsh early conditions with the fungicide seed treatment. But this had opened up some interesting stuff from the extension and research side. Is that what we found is that most growers don't know what's in their seed treatment to begin with. They don't even know that a fungicide could be part of their seed treatment. And on top of that, most growers are unaware of what fungicide options are available to them. And we kind of just highlighted some of the ones that are currently available here on this slide. And what Pooja is interested in is kind of concentrating in these water molds, the pythiums up here on the top, because traditionally we would recommend mefenoxam or this apron as a fungicide seed treatment to help. But what some colleagues out in the Pacific Northwest have been finding was that there was a lot of resistance popping up to mefenoxam seed treatments in their sweet corn. And what is available are several alternative options if we needed to switch it out and make a Virginia or a Mid-Atlantic specific seed treatment. So for Pooja's research, she's been going around. She started her this last season was her first field season. She made a tremendous effort to get around the state and really sample sweet corn fields. So she recovered Pythium isolates from 34 fields in 2024. She's amassed a huge collection. The process of getting these out of the field is no simple task either. She has a baiting assay where she has to bait them out of the soil with some corn seedlings and then isolate them and then go ahead and single hyphal tip them and get them into long-term storage. So she's been spending most of her time getting that done. She's got it in storage and she's about ready to move on to the next step, which is figuring out the species identification for all these Pythium isolates that she has collected. And so she'll utilize 2 DNA barcodes to do that, the ITS and COX2 region, and kind of figure out what Pythium isolates were there. And that's important because we know different Pythium species may behave differently to the fungicides or come with different background levels of sensitivity to any fungicide treatment that we might employ. So the second part of Pooja's project is to then look into fungicide sensitivity. So she's going to go take a look into that PA group, which includes that mefenoxam, which we would consider the standard treatment, and see if we have any resistance. And then also, since it hasn't been widely deployed here in Virginia, it's a nice opportunity to lay down some baseline data for sensitivity profiles for Picributrox, which is in a new fungicide that is a good alternative to mefenoxam and could be replaced if need be. So moving on here, the next project I'd like to highlight is a SCRI project, is part of an SCRI project that was led by Maria Velotes down at TAREC. And Maria's interested in producing faba beans in the Mid-Atlantic and planting them in the fall and growing them over the winter for spring harvest. So my group joined this project, and our task was kind of figuring out what if we were going to put in a faba bean production system, what would be the main diseases that we would be most concerned with and have to develop management plans for, because faba beans is not a widely grown crop and it would kind of be a new crop to get established here in the Mid-Atlantic. So Maria put in her plots. When Maria was putting in her plots last year, I was kind of poking around in there, and what I ended up finding was some botrytis species that I had isolated that and some symptoms that were very closely related and appeared to be chocolate spot, a disease called chocolate spot. So we decided to focus a little bit on chocolate spot, actually, before I back up here. And this year, the first plan was to kind of come in and run through some basic fungicide efficacy trials like I had done in the past, and then kind of unfold the questions from there. So we planted this big portion of the plot. And in this story, next along comes Marion, who is a master's student pictured up there on the right. And Marion gets out to the research station and she's excited to be here and it's got a lot of good energy. And so she decides to go out to the plot and see what's going on out there. And we realize that we've gotten some frost damage because we've had some cold weather. And so Marion's picking through this, frost, cold damage plot. And what do you know? She puts one of these plants with the cold damage on the microscope and she sees botrytis on there. And so now that's really got us questioning, why is botrytis there so early on? And did it come in with the frost or was it there before the frost? And so we've kind of, are going to run with this, and we don't really know, there's not really known a lot about how frost damage might interact with chocolate spot or incite it. So Marion's going to kind of pick apart this question as part of her master's degree. So she's going to start by basically just confirming that which botrytis species we have. There's 2 that could be implemented in chocolate spot, and confirm that they're actually causing the disease on the faba bean. And then she's going to take what I think is a really interesting look, and she's going to try to piece out and see if she can unwrap the puzzle of where frost damage might play in this pathogen system. If it's, inciting more chocolate spot what would happen if we had cultivars with less frost damage, and kind of try to work this out. So she's just getting started, but this is the kind of the general idea that she's going to run with. Marion's got a lot of interest in diagnostics, so her side project is she has some interest in looking in speeding up the diagnostic and being able to quickly tell between Cineraria and Fabiae, these two botrytis species, with some molecular techniques. So moving right along here on projects that the lab is doing. So we got a call out to a broccoli grower up in the Northern Neck who was saying, Doug, you got to come out here. There's something going on with my broccoli, and it's just wiping everything out. So when you hear that call as a pathologist, you pretty much drop what you're doing and you go out there. And what I ended up finding was that this grower had a field that was just covered in clubroot. And so you can see clubroot is actually a protist pathogen, and it causes these gallings on the roots of the plant, and there's swellings, and that prevents the plant from really taking up all its nutrients and growing and reaching its full potential. In this case, being able to produce a marketable broccoli head. So this was a big issue. And there's not a lot of good controls for clubroot. It's kind of one of those plant pathogen systems that's been around for a while, and no one's really nailed how to manage this pathogen. And so there are bits and pieces here. One of the things that as you can see, a theme, I always start with what the available controls are. In this case, was a set of fungicides that are labeled to help manage this disease. And so we put in some field trials with this grower cooperator, and we were just kind of really disappointed in the amount of disease control that we saw after putting these trials in the field. And this was with our spring trial. And what we learned with spring trials is that on these fungicides, the use pattern is to you get one shot with the fungicide, and you get to put it in once as a transplant drench. So when the plants are going out to the field. So we did that, and we were like, well, what we know about the biology of the pathogen that causes clubroot is that it might not be really active when it's really cold and early, and it really likes warmer, moisture, warm weathers and moisture. So what we did is plugged in some, went back and kind of took a look at what was happening soil temperature wise based on when we did the fungicide application here, that arrow on the left, versus kind of when we would expect the activity of the pathogen. And you could see there's about a 20-day plus time period between when that fungicide application goes out and when we'd really expect, disease to be present and really ramping up. So we play off this observation, and we're going to run some more trials here with this grower where we're going to put some soil sensors in the soil, and we're going to wait till the temperature hits a threshold that's conducive to the disease before we apply the fungicide, and see if we can really improve some of the management of this disease by delaying that fungicide application to a more opportune time when we would expect the pathogen to be active. And I forgot to mention, that's Kaylin Adam, who's my right-hand person on all these field trials. So Kaylin and I really scrambled to try to put in some grower cooperator trials and really take advantage of a high disease pressure scenario in a grower who was really struggling to manage disease. So we set out several different trials. I'm just highlighting one here, but Kaylin has really been instrumental in making that happen there. Kaylin also helps out and me run the work that we do for Fusarium head blight. So we have funding to work on some Fusarium head blight through the Virginia Small Grain Board, and also the U.S. Wheat and Barley Scab Initiative. We do coordinated trials with them. And this is mainly around this fungal pathogen. Fusarium head blight causes primarily quality issues in the head. It can have, in some cases, it can take a little knock on yield, but really it's about quality and these mycotoxins that are produced from the pathogen when it gets into the head. And so what we're interested in is several things. We look through fungicide efficacy as a large national group. But we also are curious about fungicide timing. What I've noticed in talking with growers is that most of our growers are applying one single product, which is Miravis Ace, because they really like the flexibility of the application window, or the perceived flexibility of this application window. But what most of the research shown to date is that you really need, there's a really tight timing that you need to hit. You need to hit right when the flowers come out, so that box in the middle, to really get some efficacy out of your fungicide application. If you're early, you really don't get a lot, and if you're late, you don't. It doesn't work as well either. And so there is this little bit of perception, although when you dig into the data, it doesn't seem to really, the trend doesn't really seem to hold, at least from all the data reports I've seen on Miravis Ace, is that these suboptimal timings, it really doesn't do that great. So part of me is kind of wondering if there was a good marketing scheme behind this product. And I'm just curious about comparing it to some of these other products that hold up just as well, or equally effective applied at the right time. And so what do they look like compared to Miravis Ace if we get these kind of off timing windows when a grower might get behind in the field or something? And so that's kind of a project that we've launched to kind of take a look at. Some other work that's funded and I'm trying to fill positions for. So if anyone on the call knows someone interested in pursuing a PhD in plant pathology, let me know. But we have this project. I have this project to improve management outcomes for Phytophthora capsicii and cucurbits. This is a disease that in some cucurbits can affect the crown and the roots, and others, and in all cucurbits affects the fruit. And so we're really looking to do with this PhD project is to do some phenotype and genotyping of fungicide resistance throughout the state, and then to kind of play on integrating strategies. So there's been some observations about age-related fruit rot resistance. So as the fruit gets older and the exocarp gets thicker, it can withstand penetration from the Phytophthora capsicii. And so I'm wondering if we can leverage on this fruit rot age-related resistance to decrease our fungicide inputs, so we don't need to have as long of a fruit rot control program, and we can decrease the number of fungicides that we use here. And then also kind of combining and playing off a potential synergistic responses between stimulating a systemic acquired resistance reaction with some of the foliar fungicides and kind of pairing them together to see if we can get bumps in disease control. And that, I think, is enough to get the PhD students started and then let them figure out a fourth chapter and come up with a project on their own. The other position, the last project I'll highlight here is one that I've kind of got going with our local snap bean growers who called me up and had me come down to watch the snap a couple times to watch the snap beans run through their electronic eye at the packing house. We're down there at 8 o'clock at night watching all the beans come in. And what they're seeing are these small spots on the beans that are highlighted here on the left, and then a symptom that plant pathologists would have referred to as russeting, which is highlighted there on the right. And what I isolated from these two sets of symptoms were two different known pathogens of snap beans. So Alternaria alternata came off the spots, and this Plectosporium tabacina came off of the russeting. And so this has really got our fresh market snap bean growers worked up. They hadn't really seen this a lot in the past, and it seems to have gotten worse and is starting to flare. And so there's a couple hypotheses that this postdoc will work on. Same postdoc will come in to do the race typing in Colletotricum. But they're going to do a pathogen survey and find out if we have more Plectosporium or Alternaria in there. It has been shown that Plectosporium is more aggressive than Alternaria. And so that could be causing us to see more of these disease symptoms in the pods. The other hypothesis that would test is if there's resistance to strobilurine fungicides, which the growers have been using a lot of, and then to also pair that with some field experiments to come up with some alternative fungicide programs to help manage the disease. The other thing the lab does is we do independent evaluations of conventional and biorational fungicides, and we work with registrants of fungicides, and we also work with the IR4 project, which is a federally funded program to help increase the availability of tools to specialty crop growers. And so we run those trials here as part of my program. And then we also run a small diagnostic lab. We take in about 65 to 75 samples. And the vision for this diagnostic lab as it's coming into place, as I'm here now going into the third season, is this is really going to become a group student project for the graduate students going through my program. So it's going to be a graduate student run plant diagnostic lab. We slowly work everyone into it and get the cohorts of students working together to process the samples and gain really some additional skills beyond their current projects that they're working on, to make them a little bit more rounded of a plant pathologist with some really good applied diagnostic skills in terms of isolation techniques, being all at ID stuff, and even just work on communication parts. And really, I've kind of set a goal for each cohort of students to validate and add to already published molecular diagnostic assays into the lab's arsenal that will just help increase our capacity as a lab to help address grower issues. With that, I'll just end on how extension is a big part of my program. So working with growers in the field, setting up grower trials, trying to get at those immediate issues that they bring us, identifying them through the diagnostic lab, but then utilizing written and electronic ways to communicate research results or disease management recommendations, a lot of one-on-one field visits. And I have pictured here, we do a disease forecasting system for our potato growers on the shore to forecast conditions that are conducive to late blight and early blight. So I just kind of threw some pictures because this just takes a big group and team of people to go through and do all this work. And I feel like something that's really special about this station is how well everyone gets along and likes to help out and work together. And so there's just a lot of people. I've mentioned, some of them throughout the presentation, but there's a lot more that are involved in making all this work happen. So this is my contact information up here. And if anyone wants to reach out to me, feel free to. And if there's any questions, I can take them, or we can move on to VJ or the next person. Or
thank you, Doug. I think Beth has a question. So VJ, why don't you start putting your slides into the Zoom while Beth asks her question.
Thank you, Mark, and thank you, Mark and Doug, for this presentation. I think as an agency, it's very important to know what is happening in our ARECs, to be aware and give a better recommendation. I know that we are having more and more pests and more strains of different pests, and we focus a lot on conventional chemicals, but we are also losing a lot of those. So is there any research that is not based on pesticide application and more, chemistry of, biology and developing new, how we call it, biology or biological control for all these diseases?
Yeah, there's a lot of biorational or biological controls out there, and they are included in a lot of the they are included in the independent evaluations that we run through. As a practitioner helping a grower survive a season, I haven't seen something that, there's few cases where there's really stuff to pull on. I didn't share. We ran through, organic fungicide options for alternaria and broccoli of 2 years of data in there, and came up with, nothing was really working. And it's not that that stuff isn't being researched where I am, and feel that I like to fit in the thing is, once stuff has become commercially available to a grower and is a potential option for them to buy and use, that's where I like to evaluate and see where its place can be and if it has any usefulness. And then also trying to combine, strategies to reduce our inputs with conventional fungicides on that side of it. But yeah, there's spots, Beth, there, and I'm interested in exploring. They just haven't popped to the surface yet in the current problems that have been brought brought to my attention on the ones we are. But I'm always open and looking. I think in every grant proposal I write, there's some version of looking at biorational products or biocontrol products and how we might be able to fit them into a system and how we continue to learn more.
Thank you, Doug.
Yep.
Thanks, Doug. All right, next for the next 10 minutes or so, we'll have Dr. Vijay Singh speaking a little bit on his weed science program here at the Eastern Shore AREC.
Yeah, hi everyone. One thing I learned, I should go before Doug next time. But I will try my best to go through my slides. And yeah, I need to run now, but I would give you kind of overview of my program, what I'm doing, and not like discussing the results. Sorry. So these are my current students and staff, 2 PhDs, 2 masters currently. And previously, like 2 master students graduated in 2023. So these students came to my program as 4 undergrad students during the last 4 or 5 years. They conducted part of their research, and some of these came through real program. My research focus is from starting from the field agronomy, cropping system-based weed management practices, and especially targeting the herbicide-resistant weeds, precision agriculture, use of drones and robotic systems, molecular biology, or studying the herbicide resistance mechanism. So right now we don't have our lab established for that purpose, but we are collaborating with several of our other colleagues, like in Mississippi State University and Pennsylvania State University, for that work. These are some of the crops I worked in the last 5 years here at Virginia Tech, corn, soybean, edamame, and we are actually evaluating several different herbicide programs and cover crops, then cole crops, potatoes, and tomatoes, sweet potatoes. So I could not place all those pictures, but yeah. And so it's quite diverse, like different crops we are working on, and especially the weed management, targeting different strategies over there. So I would start with like a herbicide resistance, since it's a main focus of my program. And we can see that there are near about more than 500 unique cases of resistance overall. And it's a global crop production challenge, no doubt. And gene flow actually can accelerate this situation, because if you have a resistant population in one field, you can easily see it spreading to the neighboring fields or rest of the region area. In Virginia, when I joined, this was the information available, and some of the cases reported, resistant cases reported, like to atrazine, some of the pigweed species, and Italian ryegrass, if we see. So Italian ryegrass was resistant to diclofop, group 1 herbicide, and also to group 2 herbicides. And we didn't have other information, but yes, there were some suspected cases, and that's from where I started working on. And these were actually our major weed species, one Italian ryegrass and also the Palmer amaranth. Now, if we talk about the Palmer amaranth, that's how it looks like, and the sphere infestation can significantly reduce the crop yield. And Palmer amaranth is not a weed of just a cotton or soybean or like one particular crop. In summers, whatever you are growing, you can see Palmer amaranth. And if it's herbicide resistant, you have very limited options. So we wanted to explore what's there in the, like in Virginia, the status of herbicide resistance. But in overall, if you see in the US, you would observe that these, like there are different groups of herbicide. ALS means like there's a group 2 herbicide. And wherever you're seeing blue means these states reported resistance to these group of herbicides. And you can see that. So it's quite concerning scenario where we don't have many options. And the PPO inhibitors, they are highlighted with the triangle, because we wanted to actually test this, because these cases were not reported by many states. So only 3 states reported the issues earlier. And we evaluated Palmer amaranth for many of these herbicide programs groups, just to indicate the resistance status. Now, wherever you are seeing these blue indicators, those were actually the locations from where we collected the samples. We contacted the extension agents. We sent our team, our technicians and students, they collected, and our collaborator, Michael Fleshner. So he sent some of those samples and whatever information he had from his earlier surveys. So it's pretty much covering all the soybean and corn growing areas. And we had the special surveys near Suffolk area just to target the cotton growers. So these are the studies results. To glyphosate, we conducted these studies, and what we observed here that we did not find any susceptible standard to compare. But if you can see the rate, we actually used 16 times of the recommended rate, and our Palmer eminence still survived the glyphosate. Same thing for fomesaphane. So that's a herbicide, the PPO herbicide or PPO inhibitor, which I indicated with the triangle. And that's what we are seeing or observing here, that we have populations in Virginia which are resistant to PPO inhibitors too. And the resistant populations were 14 times more resistant compared with the susceptible standard. Now, overall, we tested several of that, but just keeping time constraints and everything in mind, I'm just summarizing that. We collected 50 populations, and all of them are actually resistant to glyphosate. We don't have any population of Palmer amaranth which is not resistant to glyphosate. Based on this profile, whenever grower call us for weed management tactics and recommendations, we keep all of these information in mind for that specific region, and then we provide the herbicide recommendations. The other weed species which we evaluated is Italian ryegrass. Italian ryegrass was supposed to be a weed of wheat, but not anymore. Now we can see that in the like corn, soybean, and many other cropping systems. And we have observed that it is resistant to group 2 herbicides, and it is also resistant to group 1 herbicide. Pinoxidine is actually growers, they rely on this herbicide for controlling the Italian ryegrass in wheat. The only small change we are making now, instead of like using just pinoxidine, that one herbicide, you can go for the same family of herbicide like the group 1 herbicide, but with the mixture of at least 2 herbicides in that. The premix is available in the market. So these kind of recommendations we provide, and the growers are actually providing us the feedback that now they are able to control some of those resistant populations. We are examining the scenarios and continuously surveying, and we'll see how we can come up with the more solutions and alternate programs for the growers. Now, one interesting fact here that we don't have any Italian ryegrass resistant to glyphosate in Virginia. And many states have reported actually that Italian ryegrass is resistant to glyphosate too. We haven't seen any, and that's good for us because normally the growers go for burn down application before corn and soybean. So if ryegrass is normally you see in the field at that point. So that's good that they can rely on glyphosate for the burn down application during that time, and they can easily control Italian ryegrass populations. There are a lot of practical solutions to deal with the herbicide resistance, and that has been reported from Virginia and many other states previously, but not all the growers are implementing it, and not many of them implementing it. But we are suggesting that in each extension events, and also trying to evaluate some other options, what we can do and can evaluate. Like, for example, use of cover crops, because we know their emergence pattern and how we can make use of cover crops in combination with herbicides to suppress the weeds and to take control of that. With that, my students actually conducted several studies evaluating cover crops for the weed suppression. Especially one of the concern came from the growers meeting, where they indicated about the volunteer brassica issue in corn. And based on that information, we conducted several studies to evaluate the scenarios. And what we have found that the volunteer rapeseed density is not dependent on the biomass of the rapeseed, but actually depends on the termination efficiency. So if the termination efficiency would reduce by 10%, you would see the increase of 7 plants per meter square. So that's the kind of the observation we we evaluated. And then based on that, we also evaluated several different herbicide programs which can provide control of rapeseed in those programs. Some of the other, like the technologies we are working with in collaboration with the Grow scientists, a group of extension weed scientists, and Michael Fleshner is actually leading many of these projects. So we are working on the harvest weed seed control strategies like using chaff lining or the Redecop impact mill, and how effective these are. So these are non-chemical strategies to control weeds. And in this picture, it highlights how impact mill can help in controlling the weeds even without use of chemicals. So on the left side, whatever the light green color plants you are seeing, those are all weeds. And on the right side, everything is clean just because of that impact mill after 6 years. Unmanned aerial system is a focus of my area, and my program, like many students who graduated, and currently my students are utilizing machine learning algorithms and also standardizing the unmanned aerial system for spray applications. And not many programs in the US are working on that. So we are actually leading some of those projects here, and not just the efficacy studies, but also the drift management and evaluating the drift potential of that. Time is limited. I'm just moving ahead with that, just highlighting some of the results. And we have seen why drone applications in row crops and many of the other vegetable crops are effective, and why we would like to introduce to the agriculture. That what we have seen, like even with the 2 gallons per acre, 4 gallons per acre of a drone application, we can see near about 100% control compared with the backpack spray applications or the tractor-based applications, especially at the late stage applications when your crop canopy is covering your weeds. So we are just getting 75% with our backpack or the tractor spray applications. So my students are working on different algorithms to automate this process where they can identify the weeds with just few clicks within few minutes. Like the earlier, it was used to take 33 minutes to get that information. Now it just take 9 or 10 minutes to get that information. And we can easily use that information not only to detect the weeds in the field, but also for spraying with the drones for spot spray applications. Some of the other projects, like with the Grow network, we are working in collaboration with many other states, kind of building a repository of images of weeds so that would be publicly available to the people and researchers, and then they can utilize for weed management perspective. We also conducted first Mid-Atlantic spray drone workshop this last July, and people from Virginia, Delaware, Maryland, New York, Pennsylvania, and West Virginia attended it. My extension program, we are using communication extension articles, field days, farmer participatory research, and field visits to address the issues and providing recommendations to growers and extension agents. We are happy to work with them. And these are some of the pictures of our training programs. This is just to indicate the impact of our training programs and how many people have attended in the last 5 years. Near about 10,000 people attended our programs, like the total events, including the field days. These are just some of the pictures for our newsletters and what we do in extension. My weed science students, it's a consistent, they are performing well in different conferences, earning prizes, and doing great research. So I'm thankful to my team, my interns and technicians who work tirelessly at the field, and some of these sponsoring or the funding agencies who are funding my research, and also the local funding agencies like Virginia Soybean Board, Corn Board, Potato Board, and Cotton Board. With that, thank you all. I think I tried my best to cover everything in 10 minutes. Thank you.
Thank you, Vijay. You talk fast. I guess, Matthew, do we have time for any questions, or do you need to pinch it off?
Well, what I'm going to have to do, I don't want to keep people too long. If you have any questions, feel free to email Mark or Douglas or Vijay directly. I've got just a few announcements about events coming up at ARECs and Commodity Board events I'd like to make. These go through the end of March. If anyone else has some, you can throw them in the chat. But the Virginia Cotton Growers Association, for those of you who are interested in attending that meeting, will be February 18th at Paul Camp Community College in Franklin, Virginia. You can contact Dr. Hunter Frame at TAREC about that. The Virginia Peanut Production Meeting is going to be February 12th, also at Paul Camp Community College in Franklin, Virginia. You can contact Jacob Forehand at Tidewater AREC about that one. There's going to be a Beef Quality Assurance Recertification Program at Southern Piedmont. That's going to be on March 12th. You can contact Arash regarding that. And then finally, we have the Cover Crop Field Day event occurring on March 21st at Tide-Water AREC. Mark had mentioned that they are looking to schedule a Cover Crop Field Day as well on the Eastern Shore. Date to be decided, but likely the last week of March, I do believe. So with that, again, thank you everyone for attending. We appreciate your time and attention, and we will see you next month on the final Friday.
Bye, everybody.
Thank you, everyone.